Hello everyone. Today we'll be solving Cambridge International AS and A Level Biology Paper 2 Structured Question, May-June 2021, Paper 2-3. Question number 1. The Golgi body, rough endoplasmic reticulum and smooth endoplasmic reticulum form part of the internal membrane system of a cell. The membranes have a fluid mosaic structure. Figure 1.1 is a transmission electron micrograph of one area of liver cell showing a region with RER and a region with SER. Mitochondria are also visible in the image. So we can see the mitochondria. Describe the differences in structure and function between rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum, you will see that it contains ribosomes. And smooth endoplasmic reticulum shown in this particular diagram, all right, they are flattened sac, and smooth endoplasmic reticulum actually are tubular. So basically they are like tubules. So we can see the cross section of them. Rough endoplasmic reticulum is continuous with external nuclear membrane, and rough endoplasmic reticulum is more regular and layered in arrangement. As you can see, it is layered in arrangement. Rough endoplasmic reticulum produces proteins, and smooth endoplasmic reticulum produces lipids and steroids. Question number 1, part B. Phospholipids are one of the main components of membranes. Describe the structure of a phospholipid molecule. So phospholipid molecule have phosphate group and 2 fatty acid chains. The fatty acid chains are joined together with the glycerol molecule by means of ester bond. The fatty acid can be saturated or unsaturated. One function of a Golgi body is to package molecules into Golgi vesicles. A Golgi body and Golgi vesicles are not visible in figure 1.1. Describe the features other than the presence of Golgi vesicle that would help you identify a Golgi body in a transmission electron micrograph of another area of the same liver cell. So a Golgi body. A Golgi body have flattened sacs and they're layered. Each cisternae in stack is separate and contains smooth outer surface. So a typically Golgi body should look like this. Some Golgi vesicles contain secretory protein for release from the cell. Describe the sequence of events that occur following a packaging of a secretory protein into a Golgi vesicle to its release from the cell. So the vesicle, first, it will move towards the cell surface membrane by following the cytoskeleton. And then the vesicle will fuse with the cell surface membrane and the protein will be released by exocytosis. Some Golgi vesicles contain glycoproteins and glycolipids to be added to the cell surface membrane. Outline the role of glycolipids in the cell surface membrane. Glycolipid acts as self-antigen for the cellular recognition that it is part of its own cell. Question 2. Smallpox, measles, and HIV/AIDS are infectious disease caused by different viruses. These different viruses share some structural features. State one structural feature that would confirm that a pathogen is a virus. A pathogen will always contain a nucleic acid core made of either DNA or RNA, and it will have a protein coat. Question 2, part B. The virus that causes smallpox belong to a different genus to the virus that causes measles. Name the viruses that cause this disease. Smallpox. Smallpox is caused by variola virus. And measles. Measles is caused by morbillivirus. Explain why antibiotics such as penicillin cannot be used to treat measles. Penicillin only acts on bacteria and measles is caused by virus. Morbillivirus does not have a cell wall. Neither do other viruses. Morbillivirus does not have cellular structure. So that's why we cannot treat measles with antibiotics. Antibiotics may be prescribed for a person with HIV/AIDS. Suggest why antibiotics may be prescribed for a person with HIV/AIDS. HIV persons are prone to infectious diseases, and antibiotics prevents opportunistic diseases, specifically bacterial infection. Part E. Smallpox is the only infectious disease of humans that have been globally eradicated. This eradication was due mainly to successful global vaccination program. Most people who were given the vaccine gained immunity to the disease. A student correctly listed 4 reasons for the success of the global vaccination program for smallpox. These reasons are listed in figure 2.1. The virus did not mutate. A live virus closely related to smallpox was used in the vaccine. Okay, cowpox was used in the vaccine. The vaccine was freeze dried and so was thermostable. The vaccine was easy to administer, so little training was required for giving the vaccine. Okay, now the question says discuss how the reasons listed in figure 2.1 contributed to the success of eradication of the smallpox. Okay, you see, since the virus did not mutate, that means the same vaccine could be used everywhere and it could be used for many, many years to come. A person, so a person was protected for a long period of time after exposure to the vaccine. No research was required for making a new vaccine, and a live virus closely related to the smallpox virus was used in the vaccine. So you see, closely related, so same antigens and same memory cells are produced. Live virus replicates, so they give a stronger immune response, and there is no need for boosters when a live virus is injected. Table 2.1 lists 4 types of immunity. Complete each row, table 2.1, with a tick and with a cross to summarize the type of immunity gained by a person who received the smallpox vaccine. So the smallpox vaccine, since it is a live virus, active immunity is gained. It was an artificial immunity. It was not a natural immunity, and it was not a passive immunity. Question number 3. HIV protease is an enzyme composed of 2 identical polypeptide chain. Each polypeptide chain is 99 amino acid long. During translation, the amino acid are joined by peptide bond to form the polypeptide chain. Describe how a polypeptide of HIV protease is produced by the process of translation. So basically the mRNA, first of all, what it does, it attaches to the ribosome, and 2 codons are exposed to the ribosome. tRNA then carries the amino acid to the ribosome. The anticodon on tRNA then binds to the codon on mRNA. The start mRNA codon is AUG, which codes for the amino acid methionine. The second tRNA with its amino acid binds next to its first tRNA. After the peptide bond is formed, the tRNA detaches. tRNA molecules leaves the ribosome to attach to another amino acid. Part B. The synthesized polypeptide has a primary protein structure and can form a tertiary protein structure after translation. There are more bond types in the tertiary structure. Compare the peptide bond formed during translation with the type of bonds made during tertiary structure formation. Peptide bond and disulfide bonds are covalent bonds. In tertiary structure, bonds occurs between the R groups of different amino acid. Tertiary structure has hydrogen, ionic bond, hydrophobic interaction versus covalent bond, which is peptide bond in the primary structure. Peptide bond is more stable than tertiary structure bond. Peptide bond is between carboxylic acid and amino group of adjacent amino acid. Part C. Some polypeptides are translated as part of one long polyprotein chain. After translation, enzymes cut the polyprotein into separate functioning proteins. HIV protease cuts a polyprotein that has been produced within the host cells of actively replicating HIV. The separate proteins are required in the replication of the virus. Figure 3.1 shows two polypeptide chains of HIV protease form an enzyme with an active site enclosed by flaps. We can see the flap region, we can see the active site. The flap region of HIV protease is flexible. With reference to figure 3.1, suggest and explain how the mechanism of action of HIV protease can be described as an induced fit. So the flap regions open to let the flap regions, it opens to let the substrate enter. HIV protease action involves changes in the shape of active site, which is basically an induced fit to better fit the substrate and form enzyme substrate complex. Induced fit substrate is not fully complementary to the shape of active site. The change of shape to give complementary fit lowers the activation energy. The drug indinavir is one of the therapeutic drugs used in HIV antiretroviral therapy. It is similar to the polyprotein substrate of HIV protease. Suggest and explain how indinavir acts as a therapeutic drug. So indinavir acts as a competitive inhibitor and binds to the active site instead of substrate. Indinavir competes with the substrate molecule. Question number 4. Phloem sap is transported within phloem sieve tubes. Monosaccharides and disaccharides are transported as part of phloem sap, but polysaccharides are not components of phloem sap. Define the term disaccharide and polysaccharide. So disaccharide is when two monosaccharides are joined together by glycosidic bond, and a polysaccharide is when many monosaccharide joins together by glycosidic bond. Part B. Sieve tubes are composed of phloem sieve tube elements. Figure 4.1 is a diagram of one sieve tube element and its companion cell. Some of the structural features of the sieve tube element have not been included in the diagram. So we can see the phloem sieve tube element. The question says, complete figure 4.1 by drawing and labeling the structural features of the sieve tube element that have not been completed in the diagram. Part 2. A pressure gradient is a sieve tube causes the mass flow of phloem sap from the source to the sink. At the source, a decrease in water potential in the phloem sap and an increase in hydrostatic pressure of the phloem sap can be measured. Describe the events that cause each of these changes in the phloem sap. Decrease in water potential. So basically, decrease in water potential is caused by the entry of sucrose and amino acid. Increase in hydrostatic pressure. Water enters from the xylem by osmosis down water potential gradient. Question 5. Figure 5.1 shows 4 types of cell that can be seen in prepared slide of blood taken from a mammal. So we can see red blood cell here. We can see a monocyte here. Monocyte can be easily recognized by their bean-shaped nucleus. We can see neutrophil. Neutrophil can be easily recognized by their lobed nucleus. And then we can see a lymphocyte. A lymphocyte can be recognized with a round nucleus that almost covers the entirety of the cell. So the question said, write the name of the cell type in the answers line provided. We did it. Part B, during systole and diastole, the cardiac cycle, changes in blood pressure occur in the 4 chambers of the heart. Figure 5.2 shows changes in blood pressure in the left side of the heart and the aorta during one cardiac cycle. So we can see the pressure of the atria is greater than the ventricle. And at this particular point, obviously the atrioventricular valve will shut down. And the pressure of the ventricle is now increasing such that at this particular point, semilunar valve will open. At this particular point, semilunar valve will close. And at this particular point, since the pressure of the ventricle is going down below that of the pressure of the atrium, the atrioventricular valve will open back here. Now, with reference to the blood pressure changes shown in figure 5.2, state the maximum blood pressure reached in left ventricle in kilopascal. So maximum blood pressure reached, we can see, okay, so that is about 15 kilopascal. State the time at which the bicuspid or left atrioventricular valve closes. So bicuspid valve closes here. Which is 0.1 second. Part C. Chronic obstructive pulmonary disease, COPD, can cause a condition known as pulmonary hypertension. This involves an increase in systolic blood pressure in the right ventricle and in the pulmonary arteries. Figure 5.3 is a summary of some of the events that can result from COPD. COPD, chronic alveolar hypoxia, blood vessels leading to the gas exchange areas decrease in lumen diameter, or pulmonary vasoconstriction occurs, which leads to pulmonary hypertension, which then leads to alveolar capillaries. Okay, so part 1. Chronic alveolar hypoxia describes a condition where the partial pressure of oxygen in the gas exchange region of the lungs is always lower than normal. Explain how COPD leads to chronic alveolar hypoxia. Okay, inflammation of respiratory airways builds up excess mucus, which decreases the lumen of trachea, bronchus, and bronchiole. Alveoli is then able to recoil due to unable to recoil due to loss of elastic fiber. Alveolar walls then break down to form larger air sac instead of alveoli. This decreases surface area to volume ratio. So less oxygen is able to able to be exchanged in gas exchange surface, and less oxygen is able to diffuse into capillaries. Explain how the loss of alveolar capillaries affect the functioning of the lungs in a person with COPD. So, the loss of alveolar capillaries decreases the quantity of oxygen taken into blood and transported back to heart. Decreased quantity of CO2 can leave the blood into the alveolus. This increases the ventilation rate because the person need to constantly, bring in new air, and the person is unable to bring in new air. Question 6. The best time to obtain a clear image of chromosome during a mitotic cell cycle is during metaphase stage. Figure 6.1 is a scanning electron micrograph of a group of human chromosome at metaphase. The group of chromosomes shown in figure 6.1 is magnified many times. The actual width of the human chromosome between point A to B is 1400 nanometer. Calculate the magnification of scanning electron micrograph shown in figure 6.1. So the actual length here is actually 14 millimeter. So magnification is equals to image divided by actual. The image is 14 millimeter. That means we'll have to multiply it with a million in order to convert it into nanometer, and then divided by 1400 nanometer, which is the actual size. And thereby we can see that there is a total magnification of 10,000 times, 10,000x. Draw chromosome C in figure 6.1 in the space provided. Label your drawing to show the structure of the chromosome. So guys, we can see the centromere, the sister chromatid, and at the ends, we're going to see the telomere. Suggest why the metaphase stage is the best time during a mitotic cell cycle to obtain a clear image of chromosomes. So basically, at this stage, the chromosome is the most condensed, and the chromosome are all in one place. Name the stages of mitosis that immediately follows metaphase. Well, after metaphase, anaphase is the next stage. So guys, that's all for today's question paper. See you in the next class. And if you like the video, please comment in the video and subscribe to the channel for videos like this. Thank you. Bye-bye.